Hello everyone. In this video, I'm going to show you how to knock out a gene with Cas9. Not just any Cas9. In this approach, we're going to express the Cas9 and its gRNA from a plasmid that we've made with a special approach called oligoannealing cloning. So anyways, let's start at the beginning and talk about what Cas9 is and what it can do. Well, Cas9 is an enzyme, comes from prokaryotic cells, that can make double-stranded cuts in DNA. Now, there's a lot of enzymes that can do that, but what makes Cas9 special is that it associates with a special type of RNA to direct that kind of cut. So, for example, Cas9 binds to this RNA molecule over here that has two parts, a guide RNA known as gRNA and a scaffold RNA known as scRNA. Now, the scaffold simply contains a few hairpins that specifically bind to Cas9. So Cas9 doesn't bind just any RNA, it has to bind an RNA that has this scaffold in it, these hairpins over here shown in purple. Then Cas9 uses the attached guide RNA to seek out similar or complementary DNA sequences, and anytime it finds a complementary DNA sequence, it will cut it. But there's a little bit more nuance to that. So let's talk about how exactly you could use Cas9 to, for example, cut a gene, a target gene, and then mutate that gene to study its function. So anyways, what we would need for that kind of experiment would be the Cas9 protein and the gRNA scaffold RNA complex. We would also need to know the sequence of our gene. That's very important because Cas9 actually can't cut just anywhere in the genome. It can only cut on the 5' end of a PAM sequence. Now, PAM stands for protospacer adjacent motif. That's a mouthful, but all you need to know there is that the PAM sequence for Cas9 is NGG. So what I need to do first when I'm trying to cut this gene down here is I need to look for NGG sequences. So for example, here's one right here. I have an NGG, in this case the N is T, but it could be anything, A, T, C, or G. So we have TGG, that's my PAM. And Cas9 can cut upstream of that PAM there. If I want Cas9 to cut in this location, then I start at the PAM and I copy 20 bases on the 5' end of that PAM. And that will be the sequence of my guide RNA here. You can see here the PAM, TGG, and then I've copied G, C, C, G, G, so on and so forth, down to the 5' end of that gRNA. So now I have a guide RNA that will direct Cas9 to cut that specific location. So you can see there I've got my requirement in that there's a PAM right here on the 3' end of this gRNA. And I've got 100% complementarity in my guide RNA right there. Now, if that's true, then Cas9 will bind to that site and it will cut about 3 bases away from where that PAM was. So the PAM was here, it's going to cut 3 bases upstream, 1, 2, 3, in between this G and C. Now, when this happens, the cell is going to scramble to repair that cut because damage like that in the genome can lead to a lot of different problems. And usually, the cell will repair that cut without any trouble, without any mutations whatsoever. But the thing is, if the cell repairs that cut, and it's the exact same sequence as before, you can guess Cas9 is just going to come right back in, and it's going to cut that sequence again. So you have this back and forth between Cas9 and the repair enzymes in the cell, where Cas9 cuts, repair enzymes seal the cut. Back and forth, back and forth, back and forth. But eventually, the cell will make a mistake. It will either delete one of the bases there at the cut site, or it might even insert a new base there as well. And so we call that type of mutation an indel, for insertion or deletion. So let's just say in this case, that the cell mistakenly puts in a G at that cut site, and the corresponding base across from that would be a C. Now this minor change will have a dramatic impact on the expression of that gene, because what we've done here is we've inserted an additional base into that gene sequence. So all of the codons from this point forward will be shifted off by 1. And so we call that a frame shift mutation. And what that means is that we no longer have the same amino acids here on the second half of this gene. So we're most likely going to get a peptide still from this gene, but it will be a peptide that will not fold properly or will lose its function. And the earlier in the gene that you make this type of mutation, the more pronounced effect it will have on the structure and function of that gene. So there you go. In a nutshell, that's how we make mutations in genes with Cas9. All we have to do is look at the gene we're trying to mutate, find a PAM, and then down or upstream from that PAM, we copy 20 bases of sequence, and we put that into the guide RNA. And as long as we give the cell the guide RNA, scaffold RNA, and Cas9 enzyme, then Cas9 will cut the corresponding sequence in the genome. That cut will eventually be mistakenly repaired. It will get an insertion or a deletion that will mutate that gene and probably lead to its loss of function. All right, so that's how Cas9 works. Now, what do we need to actually set up one of these experiments? Well, there's actually 2 different ways you can go about this. First of all, you can get the Cas9 protein itself. You can actually buy it online. You can also get synthetic guide RNA, scaffold RNA molecules as well. And then you can just put those directly into the cell with a method something like electroporation. So there you're going to shock the cells, and that brief shock will actually create pores through which you can put your protein and the RNA. And that method is very, very effective, very efficient, but it's also pretty expensive because what you're doing here is you're paying a company, for example, to make that protein for you, then ship it. You're also paying them to make the synthetic RNA, which is relatively expensive. So there, you have a very efficient method, but it's also pretty expensive overall. So what is another way you could do this? Well, you can also transfect a plasmid into the cell that expresses the Cas9 protein and the guide RNA. So for example, you see down here in the bottom left, we have a Cas9 gRNA expression plasmid. It's got 2 expression cassettes. Now what I mean by that is that each cassette starts with a promoter, so this is a place where RNA polymerase is going to bind, and then it's going to start transcribing. In this case, the U6 promoter, RNA polymerase binds here, and then it transcribes the guide RNA and the scaffold RNA, which is shown here in purple. And then on the bottom half here, we have a different promoter, CAG, that binds RNA polymerase 2, and then it drives expression of Cas9, so here's our protein that we need. But at the same time, it also expresses in the same transcript this T2A linker, and then a fluorescent marker protein, something like GFP or mCherry. Now, the reason we put the 2A linker in the GFP here, for example, is that the 2A linker allows me to express 2 proteins simultaneously, such that every time I express Cas9, I will also express this second protein over here. So if I look at my cells and I see that they are fluorescing green from the green fluorescent protein, the GFP, then I know that those cells also are expressing Cas9. And it's reasonable to assume that they're also expressing the guide RNA as well. So the 2A linker and the fluorescent protein here give you a visual indication or confirmation that you do have successful transformation of a given cell, that they are expressing this protein and this guide RNA. So anyways, there we go. If we can make this plasmid, we don't have to pay for the recombinant protein or the synthetic RNA. Instead, all we have to do is make enough of this DNA, which is a fairly inexpensive process, to put into a cell. And once our plasmid here, plasmid just being a circular molecule of DNA, by the way, once that plasmid is in the nucleus of the cell, then it will be transcribed and translated. So we'll get the Cas9 protein, we'll get the guide RNA as well, with the scaffold attached to it. And once that's expressed inside the cell, it will migrate back into the nucleus to where it will cut the gene that we're trying to mutate. So there you go. expressing Cas9 via plasmids is a very easy, very inexpensive approach. It just takes a little bit of time. You have to actually clone your guide RNA sequence into the plasmid, and that's what we're going to talk about today. Now, I should mention one more thing, one very important distinction, which is that when we put a plasmid into the cell to express Cas9, we're going to get Cas9 expression for multiple days, probably 2 to 7 days, we'll see Cas9 expressing. You can very easily see this because GFP is expressing at the same time. So the cells will be fluorescent for about 7 days. Now during that time, we would hope that Cas9 only cuts sequences for which it is perfectly complementary, but the more time you give it inside the cell, it does actually become prone to making off-target cuts as well. So, for example, after a few days, maybe Cas9 cuts another gene that has a similar sequence to the gRNA, but it only matches 19 out of the 20 bases. That's something that definitely can happen over the course of a few days. So we're taking that risk by expressing the Cas9 gRNA complex for an extended period of time. Up here with electroporation, if I put a protein and a synthetic RNA into the cell, that's a dead-end product. It's going to go into the cell, it's going to be active for, depending on the cell, a few minutes to maybe up to an hour or so, and then it's going to get degraded and that'll be the end of it. That window of time is enough time for the Cas9 to edit the genome, but it's also a short enough period of time to where we prevent it from having the opportunity to mutate other genes. So just really quick distinction there. The method we're going to be using, which is to use an expression plasmid, is very inexpensive and it's relatively easy, but you might see mutations pop up in other sites in the genome, and you'll have to check for those later. Another thing that we're going to do in our specific approach is we're not just going to deliver one plasmid that has one gRNA. Instead, we're going to deliver 2 separate plasmids that deliver 2 different gRNAs that target different portions of our target gene. So for example, I might design one guide RNA that tells Cas9 to cut in the first exon of the gene, and then a second guide RNA that cuts in the third exon here. Now, why would I do that? It sounds like more work. Well, it is more work, but it also increases our chances of getting a mutation in this gene that actually disrupts its function. So, for example, if I only use one guide RNA, I might very well get Cas9 to cut that gene, and that cut might lead to a mutation, but I would like to point out that some codons, if you just swap out a specific base, so let's say this codon right here, TCC, if I mutate that to TCT, both of those codons encode for serine. So there I have a mutation, but the protein is still functional. So given that that is something that could happen, by using 2 gRNAs, I'm going to introduce 2 mutations, and it's more likely that one of those spots, at least one of those spots, will introduce a mutation that disrupts the protein itself. Okay, so we're doubling down here. By making 2 cuts, we are twice as likely to introduce a mutation that disrupts our protein. So just an example, some outcomes from this could be maybe I only get a mutation at site 1, maybe I only get a mutation at site 2. In a better case scenario, maybe I get mutations at both sites. Or in the best case scenario, maybe both of these guides direct Cas9 to cut at both of those sites simultaneously, and I remove an entire chunk of sequence from the DNA, from the gene. Now that would be very disruptive to the gene, and so that is highly desirable, and we're really hoping that will happen here. So you can see we've got a lot of opportunities here for Cas9 to make some kind of deleterious mutation in the gene by using a pair of gRNAs. So that's why we do it this way. Okay, so I want to introduce Cas9 and 2 different gRNAs into the cell. How am I going to do that? Well, first of all, I'm going to use 2 plasmids. One of these plasmids is going to express Cas9 attached to GFP. The other one is going to express Cas9 attached to mCherry. Now, mCherry is a red fluorescent protein, the other one being green fluorescent. So if I get a cell that fluoresces both red and green, I know that I've delivered both plasmids and I have both gRNAs. Okay, so we're going to make two different plasmids. Each of them gets a different gRNA and a different fluorescent marker. But we already have the plasmids that have the GFP and mCherry sequence in them. All we need to do is swap out the sequence that's up here. So right now there's actually no gRNA sequence in these plasmids. Instead, there's these BBS1 sites. We can use these BBS1 sites to insert the gRNA because BBS1 is a restriction enzyme. If I add BBS1 to a sample of this DNA, what will happen is it will cut the DNA in such a way that it leaves these staggered ends. We call these sticky ends, actually, because the BBS1 is cut through one backbone of the DNA and the other backbone at different locations. So this molecule is mostly double-stranded, but then BBS1 has left these single-stranded ends here. And these bases that are orphaned here, they would like to bind to something else. They would like to form hydrogen bonds with, let's say in this case, the GAGG would love to bind to a CTCC. So that gives us an opportunity to introduce our guide RNA at that location. But anyways, I'm getting ahead of myself. What the BBS1 has done here is it's cut the plasmid DNA, okay? And it's cut it at the location where we would like to introduce the guide RNA. Now, I'm glossing over a few important steps here. We do need to take that digest reaction and run it on a gel. And what we would hope to see there is we would hope to confirm that we've cut the plasmid DNA. Usually, uncut plasmid DNA appears as two bands on a gel, and we're going to see one band if the BBS1 has cut all of the DNA. So we'd like to see that, and then we take that band and extract it out of there. That gives us a pure solution of just this digested plasmid backbone. Once we have that, then we can start to design our gRNA. Now, the way we get the guide RNA sequence into this plasmid is we order synthetic oligonucleotides. Now, synthetic DNA oligonucleotides are much cheaper than synthetic RNA. I can get a synthetic oligonucleotide for about $5. Not very expensive at all. Anyways, the way I'm going to design these oligos is on the 5' end, I'm going to put a sticky end sequence. So remember we have this GAGG that's generated from BBS1. To pair with that, I put CACC on this oligo. So you can see CACC will bind to GAGG. Then after that CACC, I put my guide RNA sequence. So this is the guide that I used as the example in the previous slide. All 20 bases of it right there. But notice one very important thing. The PAM that flanks that guide RNA sequence is not here. You do not want to put the PAM sequence into the guide RNA. None of this will work if you do that. So there we go. I've got a sense oligonucleotide here, but that's just one strand of DNA. I also need the opposing strand of DNA as well. So I need another oligo. That one's going to start with the other sticky end. So here to bind GTT, I put CAA. And then I put the reverse complement of this gRNA sequence. So you can see this one starts ATTG, and this one starts TAAC. So those bases will bind to one another. If I mix these two oligonucleotides in solution, they will form a duplex. They'll bind one another, we'll have hydrogen bonding there. But notice what happens here is that I've designed these oligos to where the sticky ends are left as single strands. They have nothing to bind to. Such that if I take this duplex that I've formed and I mix it with this digested plasmid backbone, the CACC will find the GAGG. The CAA will find the GTT. They will hydrogen bond to form the plasmid, but those hydrogen bonds aren't permanent, so I also add this T4 DNA ligase enzyme with a little bit of ATP to give that enzyme some energy. And what that'll do is it'll seal the phosphodiester backbone around the oligoduplex and the digested plasmid DNA there. So that'll give me a covalent bond now that seals permanently my guide RNA sequence into the plasmid. And that's all there is to it. Well, there is one more step. After we've done this ligation reaction, we transform the plasmid into E. coli. And what that allows us to do is make a lot more of this new plasmid, pretty much. And that's all there is to it. All right? So there you go. That's what we need to do to prepare for our Cas9 knockout experiment. We need to take this plasmid right here, digest it, or pretty much cut it with BBS1. That will give us these sticky ends right here. Then we design guide RNA oligos to have sticky ends as well. Anneal those into a duplex, mix those with the plasmid, add a little T4 DNA ligase, incubate that in the refrigerator overnight, and the two parts seal together. Then we put them into bacteria, and the bacteria makes a lot of the plasmid for us. So there you go. That's everything from start to finish, how Cas9 works, and how we can express it in a cell with its guide RNA using a plasmid that was made with oligoannealing cloning.